So thus far, treatment of genetics has covered basic Mendelian genetics and also looked at two extensions to Mendel, looking at the polygenic inheritance of some traits. Some phenotypes are governed by more than one genes, and that in some cases those genes can be quantitative in nature and contributing to that phenotype. For example, the alleles that contribute to human height, for example. So we've looked at the explanation for continuous variation, that unlike Mendel's discrete variation that he observed, the continuous variation that we often observe for phenotypes in nature can be explained by polygenic inheritance. And also, unlike Mendel, who looked at basically two alleles for a given gene at a time, there can be many alleles at a given gene. And so even though every individual in a population might always bears only two alleles, if there are 4 multiple alleles, 10 alleles, hundreds of alleles, then those alleles can be found in various combinations to produce different phenotypes. So that also contributes to continuous variation. So what we'd like to pick up with now are gene interactions that, again, provide apparent but not real exceptions to Mendelian inheritance. And the first thing we're going to look at is gene interaction. And particularly, we're going to look at gene interaction with alleles of the same gene. And we've already, of course, covered dominance. We have dominant alleles and recessive alleles. But what about incomplete dominance and codominance and lethal alleles? So those are the 3 topics we're going to consider over the next few minutes. And then we'll move on to, we'll move on to gene interaction using alleles of different genes that interact. So let's look at incomplete dominance. And here's an example of incomplete dominance. Here we have a red flower whose red phenotype is dictated by being homozygous for the red allele. Let's call it the color allele, and R means red. And then there's a white phenotype for flowers, and that white phenotype is determined by being homozygous for the white allele, for the color allele. So here's the color allele, and now we have homozygosity for the white allele. And if these are inbred, we get an F1 generation that is heterozygous for the red and white alleles, and those flowers are pink. Now, Mendel never observed any type of intermediate phenotype like that, but we call this incomplete dominance, whereby the heterozygote has a phenotype that is intermediate between the two homozygotes. And naturally then, if this is a monohybrid, if these are selfed, or if these are self-cross, what we will get is one genotype which is homozygous for the red allele, one genotype which is homozygous for the white allele, and 2 genotypes that are heterozygous, reestablishing the heterozygous phenotype of being pink. So here we don't have a 3-to-1 ratio, rather we have a 1-to-2-to-1 ratio. But that does not contradict Mendelian inheritance in any way, shape, or form. It just shows us the phenomenon of incomplete dominance. And again, these genotypes 1-to-2-to-1 are homozygous red 1, 2 heterozygous, and 1 homozygous white. So that's the phenomenon of incomplete dominance. Now, before we move on, let's actually consider what could cause this, and consider a biochemical pathway that could cause incomplete dominance. So let's imagine we have a biochemical pathway that ends up in the production of red pigment. And let's imagine, for the time being, that each of these biochemical conversions, say A to C to B here, is catalyzed by an enzyme, and we'll see this again. Enzyme 2 catalyzes this, and enzyme 3 catalyzes this biochemical reaction. Well, if we imagine that the C red allele produces a functional enzyme 3, and the C white allele produces a non-functional enzyme 3. Enzyme 3. Let's start over again. Enzyme 3. That is non-functional. So then what we have is, in the case of red allele, we have plenty of enzyme 3. and this biochemical pathway produces red pigment. But if we are homozygous for the white allele, we have no enzyme, functional enzyme 3, and therefore this biochemical pathway is blocked and no red pigment is produced. So here we have a red flower and here we have a white flower. But if we are heterozygous, let's come up here and do a heterozygote. So let's do the red and white heterozygote. Here you have 1/2 the amount of functional enzyme 3. 1/2 the amount because only the red allele is producing functional enzyme 3. The white allele is producing non-functional enzyme 3. So there's half as much activity for this step in the biochemical pathway. And in some cases that produces then half the amount of red pigment in the heterozygotes. And half the amount of red pigment then is observed as a pink flower, not a red flower or a white flower. And we have an intermediate phenotype for the heterozygote compared to the two homozygotes. Now let's look at the phenomenon of codominance. And in codominance, we're really referring to cases mostly that are biochemical phenotypes. So we're looking for the presence of particular gene products in a given individual. And a classic example are the blood types found in humans, used to characterize human blood for transfusion purposes. And we're only going to consider the ABO blood type. There are other blood types as well, but let's just, for this purpose to illustrate codominance, or talk about the ABO blood group. So if you're blood type A, then you have a genotype that has the A allele homozygous or heterozygous for the A allele and the O allele. And if you are blood type B, then you are homozygous for the allele B for this blood group gene, or you are heterozygous for the B allele and the O allele. And if you are AB, then you have both the AB and the A allele and the B allele. That is, you are an AB heterozygote. And if you are the O allele, you bear the small I O allele, and you are homozygous for that small allele. So the I allele, in a way, is recessive to the others, to the A and B alleles. And this, what these alleles code for, I should say, what the A and B alleles code for are proteins on the surface of blood cells. And so if you are a blood type A, you have protein A in your blood cells, and you make antibodies against protein B because you're not making anti-B, and you don't recognize the B protein as self. So you make antibodies against protein B. If you are of the B blood type, then you have B protein on your blood cells, and you don't make anti-B antibodies, you make anti-A antibodies because you don't see those protein A's, and you would recognize protein A as non-self. Whereas if you are AB, if you're an AB heterozygote, then you see in your blood cells both the A protein and the B protein, and you don't make antibodies against either the A or B blood type proteins. Whereas if you are O, then you are homozygous for the little IO allele. Both you make antibodies against protein A and protein B because you don't see either of those proteins on your blood cells, and therefore you would recognize protein A or protein B as a foreign substance and make antibodies against them. And of course this has implications for transfusion purposes. So if we look at this a little in terms of the transfusion purposes, we see that the blood type A can receive A blood because it doesn't make A antibodies, and it can receive O blood because the O blood doesn't the O blood doesn't have any proteins that would cause the immune system of the A individuals to recognize them as foreign. And you can donate, if you are A, you can donate to blood types A and ABs because neither of these blood types have antibodies against protein A, which you have in abundance if you are type A. And the reciprocal occurs for B as well. So if you are blood type B, you can receive B and O and be fine because you're not making antibodies against B. And O doesn't have any B or A protein on it, so you won't recognize that as foreign. And you can donate, blood type B can donate to B and AB because neither B or AB has antibodies against the blood group proteins in your blood. Now, if you are AB heterozygote, you are a universal receiver. You can receive blood from anything and from any type of blood donor because you will not recognize any of the ABO blood type proteins as foreign because you have them all. You have AB. And if you're blood type O, you're not making either A or B blood type proteins. And so you can only receive O blood. If you receive A blood, you are going to be making antibodies against A. If you receive B blood, you'll be making antibodies against B, and you'll recognize that as foreign, and that will be bad for transfusion. O individuals, however, can be universal donors because they don't contain any blood type proteins that would be recognized by any of the other blood types. Now, why is this an example of codominance? Back to our main point here. It's an example of codominance precisely because you can have both phenotypes existing simultaneously. The A and the B alleles are codominant with each other. If you are homozygous A or heterozygous AO, then you're blood type A. If you're homozygous B or heterozygous BO, then you are blood type B. But if you have both the A and B alleles, if you're heterozygous for A and B, then your phenotype is both AB. It's not intermediate between A and B. It's not, there's no dominance here of B over A or A over B. We call that phenomenon when two phenotypes can exist simultaneously due to a particular genotype, a heterozygous genotype. We call that codominance, and the alleles are considered codominant. And as I've said, that usually occurs when we're talking about molecular phenotypes, the presence of particular proteins in this example. Now let's move on to another type of gene interaction where, again, we're looking at the interaction of alleles of the same gene as we have for codominance and as we have for incomplete dominance. And let's take the following example to illustrate this point. And let's see if we can figure it out. So here we have a normal Drosophila fruit fly head, and these are the lateral compound eyes consisting of about 900 single eyes called ommatidia. Here are the antennae with the feathery extensions of their sixth antennal segment here, but here's the main body of the antennae. Lots of bristles around the eyes, and we have three simple eyes on top of the head called ocelli. And then here are the mouth parts of the fly, and we're looking at the fly kind of face on. Well, a geneticist in Vancouver, Washington, actually discovered a mutation in one of their fruit fly bottles, and instead of normal antennae sprouting from or growing from the antennal sockets, there were legs instead of antennae. And this mutant was named Antennapedia, because that stands for antenna foot, so Antennapedia. So here we have legs, entire legs growing out of what should be antennal sockets. And a cross was made between this mutant fly, this Antennapedia fly, and normal wild-type flies. And we indicate the wild-type allele with just a single plus here. That means a normal allele that would cause the formation of normal antennae coming out of the head. And what was discovered was that when this cross was made between the mutant and wild-type fly is that half the offspring had the Antennapedia phenotype indicated by quotation marks, and half had the normal phenotype also indicated by quotation marks. And therefore it was assumed that the Antennapedia allele is dominant to the wild-type allele, and that the mutant fly that was discovered was heterozygous for the Antennapedia mutation. And in that way we would get, in this particular cross, we would get half Antennapedia wild-type heterozygotes, which would have the Antennapedia phenotype, and half wild-type homozygotes, which would have the wild-type phenotype. Okay, great. So what would happen then if you took these Antennapedia wild-type heterozygotes and inbred them? That is, took these offspring and inbred them. Well, you would cross, this is basically a monohybrid cross, And what was found is that in this cross, 2/3 of the progeny had the Antennapedia phenotype, and that were probably heterozygous then for Antennapedia and the wild-type allele. And 1/3 of the offspring were wild-type phenotype, that is, were homozygous for the wild-type allele. But that's not what we would expect, would it be? We would expect something different. We would expect, if this were standard Mendelian genetics, if we draw a Punnett square, we would think that these heterozygotes would produce two types of gametes. So this heterozygote and this heterozygote would each produce two types of gametes. And that when we have random fertilization, mating, we should have the following genotypes produced. We should have Antennapedia homozygotes. We should have Antennapedia heterozygotes there, and Antennapedia heterozygotes here, and then we should have homozygous wild type here. That is what we would have expected from everything we've considered thus far from Mendelian genetics, is that we would have a 3-to-1 ratio of Antennapedia flies to wild type flies. But what we got is a 2-to-1 ratio of Antennapedia flies to wild type flies. Can you think of what might cause this aberrant ratio? Does this contradict Mendel? No, it doesn't. And as you can imagine, if the defects caused by having only one Antennapedia allele, that is being heterozygous for Antennapedia, cause legs to sprout out of the head, you can imagine that the Antennapedia homozygotes might have much more severe defects, severe developmental defects. And that is in fact true. And that these genotypes die as embryos. You never see them. You never see them in the offspring here. So rest in peace, they die. And so this is a case of lethal genes. And lethal genes it's an example of gene interaction, lethal alleles, I should say. This is an example of allelic interaction, where we're talking about interaction of alleles of the same gene that in this case cause lethality and can skew our predicted 3-to-1 Mendelian ratios, for example, in this monohybrid cross. But this does not contradict Mendel. It just tells us something about the functioning of those particular alleles. And these lethal alleles cause defects so severe that homozygotes don't survive, and you therefore never see that double homozygous offspring or genotype or phenotype for that matter as well. All right. Thus far, we've considered gene interaction as considering alleles of the same gene interacting. And we've considered incomplete dominance, codominance, and the existence of lethal alleles, again, as apparent but not real exceptions to Mendelian inheritance. Everything fits nicely once you understand what's going on with these alleles. Now let's turn to gene interaction, looking at alleles of different genes. And we're actually going to take a look at the interaction of genes in the same biochemical pathway. We're going to look at recessive epistasis. So let's consider that now and consider how that, again, is an apparent but, again, not real exception to Mendelian laws. So here's an example of epistasis. Epi means upon, and stasis means to kind of to stand. So epistasis is the standing upon of one gene standing upon the actions of another gene. Consider the following situation. Here we have two strains of corn plants, that each of which have white kernels. We'll call them it's the white phenotype, the white strains. But in this case, on the right, this strain is white by virtue of being homozygous for small a alleles at the A gene. It is homozygous dominant, big B, at the B gene. Whereas this strain has a different genotype. It is homozygous big A at the A gene, and has white kernels due to being homozygous recessive at the B gene. That is, the white phenotype can be produced by homozygosity of small a alleles or by homozygosity of small b alleles. So here we have two genes, each affecting the same phenotype in a very particular way. That recessive homozygosity for either gene yields the white phenotype. So if we cross these two white strains, not knowing what I've already told you, you might expect the F1 to be all white. But in fact, the F1 are all purple. They have purple kernels of corn. And this is due to because we have heterozygosity now at the A gene and heterozygosity at the B gene. We are big A, little a, and big B, little b. And if we assume that the A and B genes exist on different chromosomes, as we'll see, they're going to assort independently, in other words, then if we inbreed these F1s, we will have independent assortment of the A and B alleles such that egg cells will be one of four possible genotypes, and pollen cells or sperm cells will be one of four possible types as well, the same four possible types. And then if we do our Punnett square fertilization, that would result from inbreeding the F1 dihybrids here, we would see the following type of situation, knowing what we know about the phenotypes produced by homozygosity for small a or homozygosity for small b. We don't get a 9:3 to 3:1 classical Mendelian ratio that we might expect in a dihybrid cross. We get a 9:7 ratio of purple to white. And this is readily understandable now by looking at the genotypes that are produced here. All you need to do to get a white phenotype is to be either homozygous for the small b allele or homozygous for the small a allele. And those situations would arise in 7/16 of the possible progeny genotypes. Whereas, if you have at least one dominant large allele at at both genes, you have at least one dominant allele, then you're going to be purple. So that would be the case in 9/16 of the possible genotypes that would arise, given independent assortment of the A and B alleles. And in a way, you could look at, you remember the 9:3:1 ratio from classical Mendelian inheritance. You can look at these 7/16 as kind of a breakout into a 3:3:1 situation, because remembering back to a regular dihybrid cross, 3/16 were had a dominant phenotype for one of the characters we were looking at and a recessive phenotype for the other. And in another three were recessive, say small d and big R, recessive for the other gene and dominant for the other one. And the one was homozygous recessive, that is D and R, homozygous recessive. So that's what's going on with these 7/16 white, is that they represent cases which, in a regular standard case that did not involve epistasis, they would have been, three of these would have been dominant for A, let's say this one, and that one would have been dominant for A, and this one, and this one would have been dominant for B and recessive for A. And then, of course, we have our double recessive here. Here, though, there are two ways to get a recessive phenotype out, and therefore we end up with 7/16 white, 7/16, in other words, recessive phenotype, and 9/16 dominant purple phenotype. And so we see this as an example of epistasis because we have these genes standing upon each other in a way that if you are homozygous for little b, it doesn't matter that you're homozygous for big A. You're still going to have the recessive little b phenotype. And if you are homozygous recessive for A, it doesn't matter what you are at the B gene, at what alleles you have. You will have the A phenotype. So that's what we mean by epistasis. This is allelic interaction where these genes stand upon each other and produce what we would at first blush see as a non-Mendelian ratio, but that we of course realize is a very Mendelian ratio once we understand the phenomenon of epistasis. And again, we can draw a little, draw an understanding of this in terms of biochemistry. Excuse me. So if we imagine we have a biochemical pathway here, say compound A is converted to compound B, which is converted to compound C. Let's say that gets converted to purple pigment. So this is a biosynthetic pathway involved in producing purple pigment. And let's, just like every other biochemical pathway, we have enzymes here. Well, let's imagine that the A gene encodes enzyme 1. And the little a gene encodes enzyme 1, which is non-functional. Call it X. And call this check. And let's say the big B allele synthesizes, so let's make it enzyme 3. And the little b allele encodes an enzyme 3 that is non-functional. So big B allele encodes a functional enzyme 3, little b encodes an enzyme which is non-functional. Well, we can immediately see what happens to this biochemical pathway, which is aimed at producing purple pigment, when we consider what the genotypes would result in. So if we are, say, big A, big B, little b heterozygote, that is a dihybrid, well, this is producing functional enzyme 1, this big A allele. And even though this little a allele is not producing functional enzyme 1, we have enough enzyme 1 around produced by the big A allele such that this step in the biochemical pathway proceeds with some efficiency. And likewise, at the B gene, we have the big B allele producing functional enzyme 3. And even though the little b allele is not producing functional enzyme 3, there's enough enzyme 3 around produced by the big B allele such that the last step in this biochemical pathway proceeds with efficiency. And we get purple pigment. So we get purple pigment here. But what if we are little a, and for illustrative purposes, let's just say big B. It doesn't really matter. So in this case, the big B alleles are both producing functional enzyme 3, and the third step in the biochemical pathway, 1, 2, 3, should proceed with good efficiency. However, because we are homozygous for the defective mutant little a allele, which produces non-functional enzyme 3, in that case, A here. So we're homozygous for the little a allele, which produces non-functional enzyme 1, then there's no functional enzyme 1 around. So this genotype will actually produce only defective enzyme 1, and this step of the biochemical pathway will be blocked. And this would lead to the accumulation of compound A because there would be no functional enzyme 1 to convert compound A to B. Likewise, you can use the same reasoning for the genotypes of, say, big A, little b. In this case, the little b alleles would produce non-functional enzyme 3, and this step in the biochemical pathway would be blocked, and you would see a big accumulation of compound C because this step would be okay, this step would be okay, but the final step would be blocked due to non-functional enzyme 3 being around, due to the lack of a large B allele, which would encode functional enzyme 3. And therefore, you would see an accumulation of compound C here, and this biochemical pathway would be blocked, and you would get no purple pigment, and you would have a white phenotype. So the white phenotype can arise because the biochemical pathway is blocked due to homozygosity of allele A, let's say it's step 1 here, or you can have the recessive white phenotype due to homozygosity, due to homozygous for the small b allele and blocking the third step of the biochemical pathway. So this is an example of epistasis, and it's an apparent but not real exception to Mendelian inheritance. And here's another slide that you can freeze this movie if you want to look at it. This indicates a biochemical pathway, a little different scheme that I've given, but essentially the same. Now, there are other examples of phenomena that seem to defy Mendelism, but do not defy Mendelism at all. And this is a very important concept, that phenotypes can be dictated by environment, not just by genotype. Yes, genotypes, thus far we've considered that phenotypes result from particular genotypes, and they do. But that's not to say that the environment doesn't interact with genotypes. So here's an example of an genetically identical individual, the same individual, let's say, Arctic fox in winter versus summer, but there's a different coat coloration. The phenotype changes in response to the environment. And you can do a thought experiment to illustrate this point too. Let's say you take, let's imagine that you have a plant here. There's a little flower on it, let's say, leaves. And you clone that plant by, plants are easily clonable by grafting, by taking a small section of a branch and cloning that plant, such that you have two genetically identical plants now growing. So these are genetically identical. But you decide to give one plenty of water and nutrients and fertilizer, and the other one you deny water and fertilizer to, or only gives very small amounts. Well, then this plant will grow much taller and be healthier than this plant. This plant will be stunted compared to this plant. So even though they're genetically identical, it's very clear that the environment that they experience can influence their phenotype dramatically. It's not the case of Mendel, who's looking at tall plants versus short plants, very discrete phenotypes. There can be a range of intermediate phenotypes if environmental effects come into play. And that is true for virtually every phenotype in nature that we see, including the phenotypes of man, of humans. The phenotypes that we observe, even in identical twins, are a combination of genotype and acted upon by environmental effects. And if we look in populations, we see variation in populations, see total variation that is due to the variation that is genetic variation that we find out there. The genes that are contributing to phenotypes and the alleles that are contributing to phenotypes vary, and that's genetic variation. But there is also environmental variation that exists. It should be an E, environmental variation that exists. And both of these contribute to the total variation that we see in natural populations. And here's another example of that. In Siamese cats, the fur grows in dark. There's dark pigmentation in cooler areas of the animal. These are the peripheral parts of the animal where blood flow is reduced, and therefore there's there's a lower temperature there. And in that case, the environment is such that the tyrosinase enzyme is active, and we get a dark pigment. But at temperatures above 33 degrees centigrade, that is the core body areas, yield inactive tyrosinase, and no pigment is produced there. So we have dark pigmentation at the extremities. And if you've ever had a Siamese cat that gets an operation and is shaved parts of its body, and it's a winter and it's cold, then a little dark patch of fur might grow in there. I've observed that in my own cat. Now, we're going to consider one other feature before moving on to genetic mapping. One other feature that seems to defy Mendel, but of course does not, and that is we need to consider the phenomenon of sex linkage. So that's what we'll be talking about in the next section of this lecture.